Hello everyone, this is Dr. Ghazal Ahmed and today we are starting a new topic. We are going to start the first in the genital ulcer series and it will be Chancroid. Chancroid is also known as ulcus mole or soft chancre. In ulcus means ulcer and mole means soft and it is in Latin. Initially, it was confused with chancre of syphilis, but it was soft, so the name soft chancre. It is caused by the bacteria Haemophilus ducreyi. In this class, we are going to learn about the history, the bacteria, and the pathogenesis of Chancroid. These two scientists, Chancroid and Augusto ducreyi, is associated with the disease Chancroid. Chancroid was the first person who in 1838 suggested that Chancroid is different from chancre. It is like chancre, but it is not exactly chancre. So the name Chancroid, but the clear description of Chancroid was given by Leon Basserio in 1852. And he suggested that a very specific feature, what is different from chancre, is autoinoculation of Chancroid. Now, Augusto ducreyi was the first person who identified the bacteria of Haemophilus ducreyi in 1890. It was isolated by Besancon and Griffin in 1900s. Moving on to epidemiology. As already told in gonorrhea, there is a epidemiological triangle consisting of agent, host and environmental factors. Host and the environmental factors remain same in all STI. Host factors include sexually active population, males are more affected than females, people having multiple partners, the homosexuals are more affected than heterosexuals, and the population having drug abuse are more affected. In environmental factors, the uneducated population and the people of low socio-economic strata are particularly more affected due to lack of knowledge. The homosexual people are more affected than the heterosexual, and the migrant workers also at high risk of STI. Now, something about the agent. The bacteria that causes Chancroid is Haemophilus ducreyi. It is a coccobacilli. Coccobacilli means that the shape of the bacteria is somewhere between exactly the bacillus and the coccus. So the word coccobacilli, and when gram stained, it stains bipolarly. Why does it stain bipolarly? It is a gram-negative bacteria. That means the structure is different from gram-positive. They have a thinner peptidoglycan layer, but this layer is concentrated more at the ends. So it gives a safety pin appearance. You can see the diagram here drawn and the bacteria showing the bipolar stain. Also, these bacteria are arranged end to end and they form parallel lines in arrangement. So they give a very beautiful appearance of like school of fish appearance or railroad track appearance. So they are arranged end to end and they come as parallel lines as drawn here. So this is railroad track and the school of fish. For better understanding, see the beauty of the arrangement. It is a gram-negative bacteria, thus it stains pink, it is non-motile, it is non-sporing, and it is a facultative anaerobes. It is a strict pathogen. For gram staining, the steps is roughly described in microscopy of gonorrhea slide. You can go and watch it. Now something about the bacillus Haemophilus ducreyi, I would like to point out, maybe previously I have written the spelling wrong of ducreyi. It is D-U-C-R-E-Y-I. So the virulence factors are pili, or fimbriae, lipopolysaccharide, outer membrane protein and hemolysin. Hemolysin is a cytotoxin. Pili of embryo helps to adhere to the epithelial cells, helps the bacteria. Now there are some differences between Haemophilus ducreyi and other Haemophilus bacteria like H. influenzae. What are those differences? The first difference is that H. ducreyi does not need factor 5 for growth. Factor 5 is NAD. It only needs factor 10, while other needs Haemophilus influenzae need both. Second is it doesn't produce hydrogen sulfide, catalase and indole. Now the last difference is it produces alkaline phosphatase. Now we'll learn the pathogenesis of Chancroid. The bacteria Haemophilus ducreyi first attaches to the epithelial cells via the pili. It contains the cytotoxin hemolysin. During intercourse, there are microabrasions in the mucosa and skin. It enters the body via this microabrasions. It is easier to enter the mucosa than the skin. Now first it colonizes extracellularly. Then it moves intracellularly. And then it has virulence factors or cytotoxins, of which 3 are main: CDT, cytolethal distending toxin. Number 2 is copper zinc superoxide dismutase. The last one is hemolysin. These 3 toxins are responsible for the tissue damage. Now, once there is tissue damage, there will be a milieu of cytokines and chemokines in the tissue, of which these 2 are important: interleukin 8, which attracts all the neutrophils to the site, and interleukin 6 that attracts the CD4 or activates the CD4 towards, and it is towards the Th1 type. These all together will now cause the papules, pustules and the ulcers, which are the clinical features of the chancroid. So this was the pathogenesis. With this we come to the end of today's class. In the next class we'll discuss the clinical features, the variants, the differentials of chancroid. Thank you for listening and do watch other videos. Thank you.